speaker is Olivia Zumsteg, and sorry if I've pronounced that fully.
Zumsteg.
Sorry.
No worries.
From Zurich, Switzerland, and thank you very much for your presentation.
Thank you very much. So, yeah, I'm Olivia Zumsteg. I'm a PhD from the crop science group at ETH in Switzerland. And today I'm going to talk a bit about my PhD project, which also involves the collaboration with the Biometris group here in Wageningen. So the title of my talk is Modeling and Prediction of Height-Related Traits, Independence on Environmental Covariates in Wheat. So I'm working with wheat. And first I'm going to talk a bit about the background, our research site, and how the data was collected. I'm going to talk about genomic prediction, about modeling with ODEs, ordinary differential equations, and then in the end, I will give an outlook. So first to the background, what's the challenge of my PhD project? So during the last couple of years, wheat yield was stagnating, as you can see here in an example from France. And in the future, there will be increasing global food demand and negative impacts from climate change, so it will get even worse. And you probably all know that yield is quite a complex trait. So we hope that phenotypic methods will help to identify traits which are related to yield. That means yield-determining traits, which are also relevant in a breeding context. And then genomic prediction hopefully helps us to speed up the whole breeding process, because measuring these traits is very time-consuming. And with genomic prediction, we could speed up everything. So what is our approach? Our approach is that we measure the plant height throughout the whole season, as you can see here in the blue line. So we measure this, and then we model the crop growth throughout the season, and then we want to extract traits which describe the plant growth, and which should, in the best case, relevant in the breeding context. So we assume that these extracted traits have also an effect on yield. And then our aim is to predict these yield-determining traits for unseen genotypes in an unseen environment by using genomic prediction and to speed up the whole breeding process. So this is a stepwise approach. We measure crop growth, we model crop growth, we extract traits based on the model crop growth and do a genomic prediction. And since this is a quite complex approach, we have this collaboration with the Biometris group here in Wageningen. So this is what our research site in Switzerland looks like. We have two different wheat fields, and they have a lot of small plots, so 2 meters by 1.5 meters. So we have two replications for each genotype. And here we have phenotypic and genetic data of around 250 different genotypes in each year, and we have 31 genotypes which were cultivated in both of the years. I'm working with the F8 generation, which come directly from the breeding program in Switzerland. And why do I work with this generation? The reason is that this is the first generation where we have yield data, and they are more or less homozygous, which is important for a genomic prediction. So they change every year, and then we measured the height in the 2 years, 2021 and 22. And then at the same time, we also have environmental sensors distributed in our whole field. So as an example, we have soil moisture sensor distributed through the field. We have a weather station next to the field and a TRH sensor. And they measure different things. So the TRH sensor measures air temperature, relative humidity. So does also the weather station. But this one also measures precipitation, irradiance, and soil temperature. And the soil moisture sensor measures the soil moisture in 9 different depths from 5 to 85 centimeters every 10 minutes. So now how do we measure plant growth throughout the whole season? So for this, we have this drone, a DJI Matrix 600, and it takes RGB images. We did around 50 flights throughout the whole season. And then we took the images and processed them into digital models using the software HISoft. And then from this, we extracted the plant height. And this is what it looks like. On the left-hand side, you can see 2 replications for a genotype, and the black points are the extracted plant height. And then we used P-splines, which were very nicely introduced by Daniela, and we model crop growth throughout the whole season. And now the question is, which traits do we extract from this model crop growth? So here we extract 3 different traits. We have the vertical lines, or the start, the time point of the start, and the stop of the stem elongation. So that means the time point when wheat starts and stops to elongate. And the third trait is the final canopy height. So that's the final height after stop of stem elongation. Then we also fitted additional 3 parameters, the so-called temperature-related traits. So here, we have the extracted height between start and stop of stem elongation. We have the temperature measured between start and stop of stem elongation. And with this, we fit a dose response curve. And here we get 3 additional fitted parameters for each genotype. So the base temperature, that means which temperature is needed for a genotype to start growing. We have the steepness of the response and the maximum growth rate at an optimal temperature. So now we have 6 extracted or fitted traits. And in this publication from Lukas Roth about the temperature response traits, you can see that they were shown to have less G-by interactions than yield. They actually had an effect on yield. And the interesting thing is that if you look at different countries in Europe, you can see that these countries differentiate in the characteristics of these traits. So based on its findings, we had the question if heritability of these 6 traits, which I just mentioned, is high enough to perform a genomic prediction. So for this, we did a 10 times 5-fold repeated cross-validation, and on top you can see the height-related traits, start and stop of stem elongation, final height, and on the bottom, the temperature response parameters. You can see we got some intermediate to high prediction accuracies in the cross-fold, in the 10 times repeated 5-fold cross-validation. But for the start of stem elongation, it was quite challenging, as you can see here, and we have differences between the 2 years. If you look at the temperature response parameters, it's even worse. So the correlation or the accuracy of the genomic prediction is quite low, and large differences between the 2 years for some of the traits. So we were wondering, why is it sometimes very low and sometimes the accuracy high? And here you can see the correlation of the same 6 traits between the 2 years of the 31 genotypes which were cultivated in the 2 years. And we can see the same pattern here. So for the traits which had a very low genomic prediction accuracy, the correlation is again very low. And we can see that we have rank changes between the genotypes. So our assumption was that maybe there are still G-by interaction included, and for the temperature response parameters here at the bottom, we could say that temperature is maybe not the only factor explaining height, and this is, possibly the case. So here, based on these findings, we realized that we really need to adjust our approach, how we model crop growth, and we fit our parameters. And this is how, when the collaboration with Biometris group started in my PhD, and we started to use ODEs, ordinary differential equations, which are able to include additional environmental covariates, such as irradiance or soil moisture content. And now a few words to these models. So at the moment, we are trying to fit these models and adjust our approach. So the first model, the irradiance model, makes the assumption that sunlight is the limiting factor. So you can see here we model the change in plant height throughout the whole season. We have a max, this is the maximum height. Here we have an intrinsic growth rate. And here in blue, you can see an additional parameter. So this is the driver function, and it describes how irradiance changes during the growing season, because we have a shorter days at the beginning of the year and longer days at the end of the season. And in this driver function, you can see we have 2 additional parameters, A and phi. And on one hand, they are environmental dependent, because, this driver function depends, the day length depends on the environment. But on the other hand, they're also genotype dependent, because different genotypes have a different response to irradiance. And this will be in the future, of course, very interesting to us when we want to perform a genomic prediction and can hopefully identify traits which differ between different genotypes. And just to give you an example, this is what it looks like. That's just an example of one genotypes in black, the measured height, and in green, the fitted irradiance ODE. Now, the soil-water model looks a bit more complicated. It makes the assumption that crop growth is limited by water, and I'm not going into detail here, but basically what it does, we have two parts here, and the first one models the change in soil-water moisture. So this is the precipitation we have in a certain environment, times the uptake by the soil, and then from this, we subtract the uptake by the crop and the runoff in the soil. So here we have, again, many additional parameters that we can fit, and some of them are, again, environment dependent, such as P here, while others, such as K, are, again, environment and genotype dependent. This is the infiltration of water into the soil, which is also affected by the root structure of the genotype. And then here, the second part, again, we fit the change in height during the season. And here we have this black part here. This is the water that goes from the soil into the crop biomass, and then we subtract the maintenance respiration. And here, an interesting parameter in the future will be, for example, C. This is the soil water uptake capacity by a crop, which is, again, genotype dependent. So here we are really interested in such traits which are genotype dependent for our genomic prediction approach. Now, this is what it looks like when we play the guitar, as Daniela would say. So we have many different kind of fits, and it's really interesting to see how the model reacts when we change the input. So at the moment, we're having lots of discussion how to integrate the real precipitation data, how to integrate the soil moisture data, which assumptions can we make, because in the end, it's quite challenging to simplify this complex world, the complex environment, into a simpler model. So an outlook, or a summary first, we can use the fitted parameters, which should hopefully be relevant in a breeding context from the growth curves, and perform a genomic prediction. And in the future, we will hopefully have more interesting traits from the ODEs and can do a genomic prediction. But then we'll have the issue that they still might depend on the environment, but then our approach will be that we hopefully can describe the dependency of these growth parameters on the environment. So thank you very much for giving me the opportunity to present this today. Thanks to the collaborators and my whole research group. Thank you.
Thank you. Well, thank you so much, Olivia, for keeping to time. This is great. So just time for a couple of, well, maybe one short question. Come on, this guitar playing is pretty impressive. I think that there could be questions. Go ahead. Just keep the answer fairly brief. Thank you.
So it's you try to follow the height growth over time, because that's easy to phenotype. But the relation between yield and height of plants in wheat is very low. So what do you think in the end it will help you to forecast which is the most productive genotype?
So in the end, we just try to fit parameters which hopefully should have a correlation to yield. So in the publication which I showed from Lukas Roth, he could show that these temperature response parameters actually have an effect on yield. And the idea is that since yield is quite complex and has a lot of GBA interactions, we hopefully can break it down and fit traits which have a lower GBA interaction. That would be the aim.
Okay, thanks.
Okay, thank you. And thank you very much, Olivia. Thanks again.